So our speaker for today is Dr. Serena Nik-Zainal. She's a CRUK Advanced Clinician Scientist and an Honorary Consultant in Clinical Genetics. Dr. Nik-Zainal qualified in medicine from the University of Cambridge, Murray Edwards College in 2000 on a scholarship from Malaysia. She undertook a PhD at the Wellcome Sanger Institute in 2009, exploring breast cancer using whole genome sequencing. Her detailed analysis revealed imprints left by the mutagenic processes that occurred during cancer development. Dr. Nik-Zainal moved to the University of Cambridge in order to accelerate the translation of her genomic expertise towards clinical applications and to further her work in the physiological mechanisms underpinning mutagenesis. In 2019, Dr. Nik-Zainal was awarded the prestigious Dr. Josef Steiner Cancer Research Prize for her work in cancer genomics. And most recently, I've been told by Professor Chris Huang, who is also from Murray Edwards College, that Dr. Nik-Zainal has just been elected as Honorary Fellow at Murray Edwards. So if you're from Murray Edwards, please join me to congratulate Dr. Nik-Zainal. And without further ado, let us welcome Dr. Serena Nik-Zainal to deliver this talk, DNA, Inspiring Building Blocks That Made Me a Learner in Perpetuity. Dr. Nik-Zainal, please.
Thank you so much, Prem, for the very kind introduction and for inviting me to come and give this talk today. I'm feeling really, really honored to have this opportunity. So I thought I'll tell you a little bit about my research work and starting from pretty much the beginning. I do apologize if some of you already know some of this stuff, but I think I probably need to give a little bit of background before I launch into too much of the detail. I'll try not to make it too heavy, try to keep it light. So I thought I'd just start by saying that I train in medicine, as you heard from Bram, but I do remember a moment during my registrar training to become a specialist when I got the opportunity to see some cells under a microscope, and there's still something about seeing the cells in mitosis that was still really inspiring, or it's sort of beckoning. It's just very exciting to be able to see it, and I know it seems really silly, but I think there was always this grain of interest in science and in chromosomes and in DNA, and I've always had that. They say that you cannot change what you are, only what you do. And so I'm going to start with how we, when we were first conceived, would have had one copy of the human genome, the genome that we inherited from our parents. And the single copy has got to replicate again and again and again. So you start off with your 23 pairs of chromosomes. In this case, this is a chromosome set for a woman. You've got 23 pairs of chromosomes, and that has to replicate again and again and again. And actually, over time, you can acquire changes, genetic changes or mutations that happen on all sorts of scales. So, in fact, by the time you're an early adult, young adult, you probably have a few chromosomal abnormalities and probably quite a few mutations already in your genome. At some point, a cell may acquire a genetic change which we call driver mutations. So these are a mutation that happens in an important gene and that confers some kind of growth or selective advantage to the cell so that it can now come to dominate an area. Sometimes you acquire more than one driver mutation, you acquire a couple, and the cells become a little bit more awry, and eventually it turns into a frank malignancy, and you can see that the chromosomal complement here has changed remarkably. So a cancer has always been thought to be a disease of the genome. And as you can see here, what I'm trying to show you is that it's come from a single cell that has eventually turned into a cancer. So it's a clonal outgrowth from one cell that has gone horribly awry, but now it can escape its compartments and wreak havoc on the person. So cancer is a disease of the genome. And that was the dogma. That was what I was taught when I was in medical school, when I was a student, even when I was a PhD student in my 30s. Cancer was a disease of the genome. And I hope by the end of this lecture, you'll see that I'm not terribly sure that is the case anymore. And so I think one of the points that I'm trying to communicate even in the title is that I'm always learning, right? Every time I think I understand something, it changes again a little bit more. So it's kind of a humbling process, this doing science in medicine. So one of the things that happened was the advance in DNA sequencing, the ability to read the genome. And the speed and the scale of sequencing just went up enormously. So in the time that I was in medical school and through my years as a trainee medic and as a young consultant, the speed and scale of sequencing increased by orders of magnitude. It was this fast. The human genome, of course, we know is 3,000 million base pairs. And when the Human Genome Project was first conceived as an idea, this still took 10 years to sequence a genome. It took thousands of scientists and $3 billion thereabouts to sequence one human genome. But today, we can read the entire human genome. We can sequence a whole genome in just one day. And I have to say, this has happened really quite recently. When I started my PhD in 2009, it still took us months to get a human genome. Today, we can do it in one day. And when I say we, I don't mean me necessarily. I mean the people who come up with the idea of how to use the chemistry of sequencing. But that means that the ability to study diseases like cancer, which we know have lots and lots of mutations, becomes instantly amenable. And so this idea of looking for these driver mutations that I mentioned at the start, it became really relatively easy. We were trying to find driver mutations like this set of 4 that I'm showing here. These are just examples. The reason people were looking for driver mutations was because they become targets for therapeutic intervention. So here are a couple of examples of drugs that have been designed specifically to target these driver mutations. But apart from the driver mutations that are in the cancer, as you saw in the first few slides, that you also acquire many, many additional mutations throughout the genome. And so this is just like a cartoon over time, going from the age of 0 to 70. And the thing on the left is what we call a phylogenetic tree, the evolutionary tree of a cancer. The stars and dots represent the driver mutations, usually 1, 5, maybe 10 driver mutations per cancer genome. The reality is you've got many thousands of additional mutations that are not called driver mutations. They are called passenger mutations, and they're believed to be bystander events, just damage to the genome that occurs because as a cell turns from being a cancer, from a normal cell to a cancer cell, they acquire all these passenger mutations. So some of these mutations may actually be perfectly normal because, hydrolysis and deamination are processes that just happen. Our cells have got water in them, our cells have oxygen in them, and these biochemical processes just happen naturally. One may be a user of a sunbed or a smoker and expose the DNA in our cells to these exogenous mutagens. We could have little bursts of mutagenic activity or a DNA replication or DNA repair pathway that's gone awry. So DNA repair pathways are there usually to mitigate DNA damage. You're usually there to try to fix any DNA damage. But you can have a DNA repair pathway that's gone awry. So each of these different mutational processes can leave its characteristic imprint or a signature on the genome. When you sequence a cancer genome, when you read the whole cancer genome, what you get is a final portrait. It's a composite of all those signatures added together. And it's a function of the amount of the exposure as well. So if you've done a lot of smoking, we're going to see a lot of smoking patterns, smoking signatures in your cancer genome. So it may look like a random collection when you sequence this genome here, but actually you can use mathematical methods to extract the signatures, to identify these different patterns. If you have many different tumors, you can identify the patterns that are the same or shared between different tumors, and you can even quantify them. So you can identify the pink rectangles in this diagram. You can even identify the arrows, the size of those arrows. So as I mentioned, there's many thousands of passenger mutations. So just to put that in context, just so that we understand that, because you're there going, okay, I'm not completely sure that I understood what she was talking about there. I sort of, when I try to explain this, I sort of say that, it's like looking at a beach full of footprints, which looks really random and nonsensical, but actually there are patterns in the footprints in the sand. So If you were to look at footprints from shoes from humans, by studying those footprints, you can identify whether it's a child or an adult. If you examine the length of the stride, you can have an indication of how tall a person is. So you can get quite a lot of information about how many people are walking across the beach. You can even tell an animal from a human. You can tell different animals. Usually, I try to ask the audience what they think this one is, but because this is not interactive, I won't be able to hear it. But yes, I'll let you guess. And then this one is another animal, which is a monitor lizard. So the one at the top is a turtle. It's a giant turtle that's come to lay its eggs on the beach. And the one at the bottom is a monitor lizard, and monitor lizards come along and they eat those turtle eggs. So by studying the footprints on the beach, you can get a sense of what's going on this beach, the drama that's occurred on the beach. And so the same thing as looking at the mutation patterns in a cancer, you can get a sense of the mutational processes that have been occurring in the cancer as it turned from being a normal cell to a cancer cell. Right, okay, so when we started doing whole genome sequencing in cancers, we had to present these patterns in some way. So there are 6 main classes of mutations, as shown in the different colors here. You might ask me why there's 6 and not 12, because a C to A in a double helix is the same thing as a G to T, because C to Gs are partnered and A to Ts are partnered. A C to G is the same thing as a G to C, a C to T is the same thing as a G to A, etc., etc. So there's basically 6 main classes, and we can look at some of them in more detail. So let's look at C to Ts in more detail. I've just blown it up here so that you can see it more clearly. You can also take the 5' and 3' base into account. So there are 4 possibilities, each 5' and 3', right? ACGT. So 4 by 4 is 16. 16 multiplied by 6 gives you 96 channels. And so the signature, the mutation pattern, is the whole 96 element pattern. The whole thing is called the signature, not just the tall bars. But I will point out the tall bars because they give you some idea of what the signature is about. So in this case, the tall bars, C to Ts, are occurring at CPGs. And this is a well-known mutational process. It's called deamination of methylcytosine. So if you have a C next door to a G, the C is often methylated. So methyl Cs often deaminate into T-thymines. And this is a well-known process, well described by other people before we even showed this in whole genome sequencing. Here is another deamination process, another endogenous intrinsic process happening in all our normal cells. And it can result in 2 distinct signatures called signatures 2 and 13. So these are just things that happen in your cells. They're intrinsic. Here are some examples of mutational signatures that occur due to environmental exposure. So the top pattern characterized by these tall red bars, C to T bars. This is very classic. There's 4 here and another 4 here. This is UV damage. You find this in malignant melanomas. This pattern here, signature 4 with the tall blue bars, our C to A mutations are due to tobacco smoke in lung cancers of people who've been exposed. And then this pattern here with the tall T to A bars is a well-described pattern that's been seen in liver and kidney cancers of people who have ingested ursolithic acid. Ursolithic acid is endemic in some parts of the world. It's also present in Chinese herbal medicine. So this is a pattern that's seen in some cancers in certain parts of the world. So that's just to give you a flavor of the kinds of signatures there are. There's actually about 49 or 50 of them now. I'm not going to show them all. I just want to communicate to you that there are these mutational signatures. We identify them in cancers. Some of them are intrinsic. They come from within your cells. Some are exogenous or environmental. And then to give you a sense of what this looks like in individual cancers, we're going to look at 4 different tumor types here. So there's breast cancer, cervical cancer, CLL is a kind of leukemia, and lung cancer. And I've chosen these 4 for a reason. Each bar here represents a patient's tumor, and the colors represent the different signatures that are present in each person's cancer. And you can see that everyone's got sort of different numbers of mutations. Everyone's got slightly different colors. The first thing I want to say is that deamination of methyl cytosine signature that I mentioned right at the start is the green ones. And the green signature you can see is present in nearly every single sample of every single tumor type. And that's because deamination of methyl cytosines is normal. It's common. It's happening in all your normal cells as you sit here listening to me. Then there's the pink signatures, which are due to this enzyme called ApoBec, also a natural deamination process. But note that there are some patients who've got really high numbers of mutations. We call these a hypermutative phenotype. And so there is this diversity in the rate of mutagenesis of some intrinsic signatures. Then there's the blue signatures, which is, or the light blue signature, which is signature 4 due to tobacco smoke. You don't see it in any other tumor type. You see it in lung cancers or other oropharyngeal cancers in smokers who have been exposed to tobacco smoke. So this is just to give you a sense that there are some signatures that are present in many tumor types. Some signatures can have quite a wide variation of signatures of rates, and then some signatures are due to environmental exposures, but very specific. You'll see them only in tissues that have been exposed. Okay, so at this stage, you know, this was at the end of my PhD. It was really thrilling to have done all this whole genome sequencing work to show all these patterns. We had big papers. It was very, very exciting. And this is how I felt about the whole thing. You know, there were a lot of naysayers at the time. And, you know, they were sort of saying, well, whole genome sequencing is all very well. But first, it's terribly expensive. And second, every cancer has got a different set of drivers or signatures here. Every tumor is different to each other. No two tumors are alike. No two tumors have the same drivers or signatures. So how do you make any sense out of this data? And I hope to show you that actually, even though no two cancers share the same mutations and it may feel like a really impossible task, actually, if you take a holistic approach, you can interpret whole cancer genomes. I'm going to show them some of these whole cancer genomes to you. So these are two patients. They're two different patients, and all their whole genome data is in this plot. These are called CRUK plots. I'm going to orientate you. The woman on the left is 47-year-old, and the woman on the right is an 80-year-old. So on the outside most ring, the outermost ring, it's the chromosomal ideogram. And so we're going clockwise, chromosomes 1, 2, 3, 4, or 5, all the way around to X and Y. Then the next layer in, these dots, are substitutions, single base changes. The dots are colored according to the different mutation classes. Remember, I talked about the signatures. You can quantify the signatures. And these two patients have both mainly got signatures 1 and 5, signatures 1 and 5. They've got a total of about 2,000 mutations each. The next circle in are small insertions or deletions. And this is when there are a few bases that are removed or inserted. So in green are deletions, in red are insertions. In red are deletions, in green are insertions. And here's the graph of it all when it's being aggregated together. So not very many, 205, 139. The biggest bar is the green bar. We go one more layer in, and this tells you about the number of copies there are of chromosomes. Remember, we should be diploid, unless apart from the sex chromosomes. And anything in green basically is an excess of chromosome, and anything that's in pink is too little chromosome. So in this case, the arrow is pointing to a green section on the long arm of chromosome 1, and both of the patients have the same thing. So they've got a green section that is increased, so they've got a gain of the long arm of chromosome 1, 1q. They've both also got losses at chromosome 16. So this is the long arm of chromosome 16 is lost in both of these patients. So although they are two different patients, they have similar chromosomal copy number changes between them. This is a well-known phenomenon in breast cancer. And then if we go inside again, here there are lines which represent structural variation. That's when your sugar phosphate backbone is broken and you go off and you form a translocation or a rearrangement with another part of the human genome. So these are the main classes of mutations in whole genomes. And although there's not a single mutation in common between these two patients, overall, when you step back and you look at these profiles, they're rather similar. They're both rather sparse. They mainly have signatures 1 and 5. They mainly have this big green bar. They don't have many rearrangements. They've got a 1q gain, 16q loss. They do have some similar drivers. So this one is PIK3CA. This is a driver PIK3CA. This is a driver GASA3. She doesn't have a GASA3 driver. So no mutations in common. but overall, the profile is rather similar. I think you'll agree it looks very different to these 3 women who have got very many mutations on the outside. And you can see these dots are mainly red and green because they have a particular signature. They've also got these circles are nearly solid. These are indels. They've got lots and lots of small deletions and insertions, and the graphs here show you that they are in the thousands. And these deletions are occurring of a particular variety. They're occurring at polynucleotide repeat track. There is a repetitive sequence in the genome. And this is classical of a repair pathway that has gone awry, which is a mismatch repair pathway. So these are tumors that have got mutations in a gene called MLH1. MLH1 is known to be a mismatch repair gene. And these tumors would potentially be sensitive to a very particular drug, immunotherapy agents or checkpoint inhibitors. And then these are 3 patients that have had exposure to ursolithic acid. And what's interesting is none of them are aware that they've had exposure to ursolithic acid. So the tumors, the cancer genomes, can tell you stuff that you wouldn't necessarily have known. So ursolithic acid is something that people in Asia would have some exposure to in some parts of the Balkan states, but not necessarily something you'd see much in the UK or the US. So these 3 patients had no idea that they had this exposure. And then these are tumors from patients who have inherited a genetic defect that gives them early onset breast cancer. It's called a gene called BRCA1. And I hope you can see that they look very different to any of the previous ones. They have got a very different sort of pattern on the outside. They've got a lot of black dots here. They have a lot of pink going around here. These are copy number losses. They've lost a copy of one of their chromosomes throughout the genome. They've got a lot of these green lines, which are rearrangements beautifully dispersed throughout the genome. This is classic BRCA1 mutation appearance, whole genome appearance. They're really very easy to distinguish from the previous cancers. So reading a cancer genome can tell us what has caused a person's cancer. And this could help us to determine what are the most appropriate treatments for each person's cancer. So I like to think, I guess the way of looking at it is, of all these signatures, of all these patterns, of all the footprints that we see, some of them may be perfectly benign and actually not very interesting or important, but there will be some that are really important. So this one, the monitorates, it's a really important one because they come along and they eat the turtle eggs. So this is something that we need to be aware of and to try to target. So this is my mom, and my previous picture was of my daughter having a nice time on holiday, enjoying life and celebrating her holiday. And this is my mom, who is a Chinese lady who's pretty tough. Where I was doing all this mutational signature work and very pleased with myself. And she was like, yes, that's all very interesting, but how is it helpful to mankind? And I think she's been a real grounding force and a person that, I've tried to sort of live up to the standards here to try to find something useful to do with these mutational signatures, I guess. This is also her. At this point in time, she has been diagnosed with metastatic cancer, sadly, and this was the last holiday she had with us to Malta. And even though she had her cane, which you can see there, you can see her climbing the walls to look over the ramparts because she is that sort of person. She's always one to kind of explore further. So the next few slides are going to be all about how we've tried to turn these mutational signatures into algorithms that help us to get some clinical utility for patients. And as you would have seen earlier, that the whole genome plots, the CRUK plots from breast cancer, this was one of those patients earlier, just wanted to just reinforce this point that it looks very different to this tumor, which you also saw earlier. This is from a woman with a BRCA1 mutation. So I'm just going to highlight some of the patterns that are different between them. So the woman on the right has got largely signatures 3 and 8, unlike the lady on the left-hand side. These are substitution patterns. She also has different indel patterns. Indel signatures show that she has got deletions, this burgundy bar, deletions with microhomology. This is a little bit of sequence that's very similar to each other at a deletion junction. She also has 300 rearrangements, and they're all these green tandem duplications beautifully dispersed throughout the genome. She also has got copy number losses throughout the genome, so these pink stuff throughout the genome. So there's 1, 2, 3, at least 4 or 5, in fact, different varieties of mutation patterns that are different to this other tumor. And so that's what we use in our machine learning methods. We identified a whole bunch of women who had these BRCA mutated cancers And then we contrasted them to women who just had standard breast cancers to see whether we could create an algorithm that could help to predict the state of BRCA loss. This state of BRCA loss is sometimes called BRCAness. BRCA are involved in a repair pathway called homologous recombination, or HR. It's a really important pathway that helps to maintain the genome in a pristine state. And so the reason why we were trying to create this algorithm was because there had been hints that there were more tumors that had BRCAness, and potentially these tumors could be sensitive to the same drugs that had been designed for women with BRCA1 and BRCA2 mutations. So those drugs are things like platinums and PARP inhibitors. And maybe if we could create an algorithm to identify more patients, perhaps more patients could be sensitive to these drugs. So the algorithm is called HR Detect. And we applied it onto a cohort of 560 patients. So you've got 560 patients on this axis, and on this axis here, you've got the score. A high score predicts BRCAness and a low score predicts not. So remember, this is based on the signatures. All we're doing is running an algorithm to look at the signatures and to report back whether it thinks that the sample has got HR deficiency. So we found 124 patients were above our cutoff of 0.7. So that's quite a lot. That's 22% of the cohort. We weren't really expecting that. We were expecting maybe 1-5%, not 22% of the cohort. That's 1 in 5 breast cancers with a HR deficiency, a BRCAness phenotype. So we took a closer look at that group. And we did find that 22 women that we knew had genetic BRCA1 and BRCA2 losses. They had inherited this, and we knew them. They were our positive controls. But interestingly, we found additional cancers that also had the BRCAness. So we had 32 patients who had new diagnosis of having inherited BRCA1 and BRCA2 mutations. This opened a can of worms, actually. So this had clinical implications for their families. They could have screening, etc., and they could have things like bilateral mastectomies and remove their ovaries as a result. So this had a significant impact on the families. There were 22 women who had acquired BRCA defects. So they were not born with BRCA defects, but they acquired it relatively early in life. And then there were 47 cases, so about a third of the cohort, where we could not find a BRCA1 or BRCA2 genetic cause for the signatures that we were seeing. And so, you know, at this stage, we have got a algorithm, a clinical algorithm. It has been trained on our data set, and we're finding all these additional patients. And that's all very well. At the moment, it's predicting biology. Does it predict clinical outcome? And to figure that out, we have to apply it into population studies or into clinical trials. So I was very fortunate to work with, collaboratively with these two men, Johan Staff and Åkerbo from Sweden. And the wonderful thing about the Northern Europeans is that they have these amazing collections of clinical information, treatment, outcome data since whenever. And they had been collecting samples, breast cancer samples, from all of these little hospitals. These are just district hospitals in the whole of the south of Sweden. Every woman with a breast cancer was recruited to this study called ScanB without exception. They had an 85% uptake. By the time we started talking to each other, they had like 11,000 samples. And because they have treatment and outcome data, I can now test my algorithm to find out whether my algorithm predicts anything or not. And so that was great. 10,000 samples is a lot, and it costs far too much, so I couldn't do 10,000 samples. And there's no point anyway, because a lot of breast cancers are cured. What I want to do is work on a cohort of tumors which are, you know, are an area of unmet clinical need. So tumors that are still have an aggressive phenotype that if you were diagnosed with, you would still generally have a relatively poor outcome. And so we worked on a very particular kind of breast cancer called triple negative breast cancer, or TNBCs. And we sequenced every TNBC in south of Sweden between 2010 and 2015. So we have a clear window, and that has given us enough time for relapses to occur or deaths to occur. So now we also have outcome data for these patients. So here are the results. We found that 58.6%, so more than half the patients, had HR deficient tumors. That's a lot. We kind of expected that in TNBC. We knew that TNBC tumors tended to have a lot of HR deficiency. But the question is, does it predict outcome in any way? And so this is called the Kaplan-Meier plot. And what you have on the y-axis, on the vertical axis, is invasive disease-free survival, so how much time they have survived without any further recurrence of the disease. And on the horizontal axis, you've got the time in years. And so we've separated the patients into, in black are HR detect high cases, so these patients on this side, and then in gray are the patients with low scores. And what you can see here is that the black line stays up, high up, because many more patients are surviving over this length of time. So this is a good outcome in black. So the point here is that HR Detect was able to prognosticate patients with tumors that had high HR Detect scores in the black line had better outcomes than patients who had low HR Detect score. And this is on standard of care treatment in Sweden. So that's all very well. So if you get a high HR Detect score, basically you do pretty well on current treatment or treatment in Sweden at that time. But in this cohort of patients who have high HR Detect scores, you could ask the question, could you just find the mutation in BRCA1 and BRCA2? And the answer is we found it in about 2/3 of the patients again, not in a third. So we could see the signatures and the patterns. We could tell the tumor had BRCAness, but we could not find the genetic or epigenetic defect. So epigenetic is when you turn off the gene in ways that are not genetic. So we just couldn't find a cause for the patterns, but we could see the patterns. But that's important because it means that the signatures are telling you something that you wouldn't otherwise be able to detect. And so we looked at all of these patients in this black line here. We split them into the patients where we could identify the genetic defects and the ones that we couldn't. So in black are the ones that we can, and in gray are the ones that we can't. And you can see that there is really no significant difference between those two lines. In other words, if you take the signature approach with this HR Detect approach, you are able to discriminate tumors that have an improved clinical response to standard of care therapy, even when you cannot find the genetic cause for the patterns that you're seeing. So the signatures can prognosticate, they can help you identify patients that could respond to treatment. So a couple of notable points here, which is, although I kind of focused on the algorithm, the reality is to do the algorithm, you had to sequence the whole genome. And remember, I've just showed you a whole bunch of whole genomes. You can see all sorts of other information there. So the first thing to say is that if you did a whole genome, or rather, if you didn't do a whole genome, you would miss all the patients here in gray. You just wouldn't spot these patients that have the pattern, but where you cannot find a genetic defect, because currently people still think that whole genomes is too expensive. So they just sequence some genes, and BRCA1 and BRCA2 are some of the genes that they sequence, and people will argue there's no need to do a whole genome, just sequence the one little gene. But the point here is that if you took that approach, you would miss this 30% of cases who actually would respond. The second thing to say is that currently, when you do clinical trials to test a drug, people do use genetic information, but they use it in a very binary way. They look at a mutation and then they say whether with the mutation you go into the arm to test the drug or not. And so just to give you a sense, these are driver mutations. Here's a list of driver mutations across this cohort of triple negative breast cancers. I'm going to draw your attention to one particular one called PIK3CA, because PIK3CA mutations are often used or sometimes used as a way to stratify patients in clinical trials. But as you can see, PIK3CA mutations are distributed throughout this cohort of patients. And actually, there is a priori information, there is background information. If you bothered to look at the whole genome, you might find out that actually some of these patients would have been stratified because they had the PIK3CA mutation, would have responded just because they had a high HR Detect score anyway. So sometimes I sort of stop and I go, I still can't quite fathom how we use a single data point, one driver to make a decision about what treatment a patient should have, when there are all these other mutations in the genome that can give you information about the genome already. We just don't use it. So, but actually this has been a really, for me as a scientist and as a clinician, it's actually been something that I've been banging my head against, because this is the mindset. Everyone uses genomes, or everyone uses genetic information in quite a binary way. because everybody else is doing it. I don't think, there's any malice in people. That's just been the way people have worked with genomic information. Don't just do something because everybody else is doing it, I think would be the point there. I know it's the norm, but there is a mindset shift that needs to happen in terms of using all genomic information. So then the last point to make here is that, in this cohort, which I pointed out earlier, had really poor outcomes. They have 4% of this cohort have a DNA repair deficiency called mismatch repair deficiency based on mutational signatures. So they have these additional features, they have these additional patterns that tell you what is wrong with the tumors, but they just would never get the treatment usually because nobody looks for them. So mismatch repair deficient tumors tend to be sensitive to immunotherapies or checkpoint inhibitors, but these are breast cancers. In breast cancer, we do not look for mismatch repair deficiency because we think it's too rare. And so these patients just wouldn't get this drug because it's just not an option. We just don't look for them. I hope I've sort of given you a little bit of insight into the sort of process that we've gone through, which is we started with mutational signatures as a nice academic thing, and then we developed an algorithm, and now we've tested the algorithm in a big population-based study. There has been other work. I won't go into it. The point is that you can take an academic thing and then develop clinical tools with it, and then try to apply it, validate it in population studies as you head towards clinical implementation. Now, a lot of the work, which is largely analytical, is supported by experimental work. In the interest of time, I won't go through all of it, but I will just highlight that we have done some of these studies just to make sure that we're not talking nonsense with the analysis, and actually we can reproduce some of these in a dish. So we're working with human cells, and we're either treating them with lots of different environmental mutagens that are present around us, and also we're taking the same cells and we're knocking out different genes that we believe to be important in maintaining the genome. And so we're doing this in a very systematic way. We're trying to create a reference set of mutational signatures generated under experimentally controlled conditions. So now that in the future, when you sequence your genome and you find your patterns, you can refer to the reference database of signatures and say what's in your tumors. And just to give you a little bit of insight into what some of that looks like, this was a study done with Jill Kukab in King's and David Phillips and Shui-Ching Zhu, who is a fantastic postdoctoral fellow in my lab. We treated we used a whole bunch of different compounds that are present in the environment. There's 13 different families here, 125 different treatments, 79 different agents. But this is just to show you that we had a whole bunch of controls as well, that you actually have a low level of mutagenesis, a low level of background accumulation of mutations in the controls. But in treated cells, you get a huge number of mutations and really quite a lot of them. And the asterisks are all clinically significant changes in sort of mutation burden. You don't just get substitution changes, you get indels and double subs as well. And so this was just a very interesting study demonstrating that we can reproduce, so we can validate a lot of the signatures that we've identified in human cancer. So this is already published, and this was a very systematic study. We identified 41 substitution patterns, 6 double sub signatures, and 8 indel signatures as well through this study. We also did the study where we knocked out different DNA repair genes, as I mentioned earlier. So here's just a sort of a snapshot of all the different genes that we managed to knock out. This was a piece of work that's been done in collaboration with Bill Scans at the JAX and Joe Jurikny in Zurich. I don't have enough time to go into all the details, but I just wanted to emphasize that apart from knocking out these genes, we also had patients that naturally had some of these DNA repair defects. So just to back up our findings, when you knock out a gene, if you get a pattern, if you also have people who are naturally occurring knockouts, as it were, do they have the patterns as well? And the answer is they do. So this is an example of children who have something called constitutional mismatch repair deficiency. They have a failing of the mismatch repair pathway in all their cells, and they have characteristic features. They get these cafe au lait patches on their skin, and they get brain tumors. They often die in the first decade of life. And we managed to make some stem cells or pluripotent stem cells from these patients. And sure enough, we have a mutational pattern. These are 4 different genes, which are quite specific. So these 3 patterns, these 3 genes produce the same pattern, and this gene produces a slightly different pattern. But I think the thing I want to try to communicate here is that these are not cancer cells. All the experimental work that we have done has been in normal cells. They've been in normal stem cells. So that sort of made us stop for a moment, because all the work we've done has been in cancer, and we have kind of always thought of mutagenesis as being something that is big and it happens a lot in cancer. But in fact, all these experiments, and we produce thousands of mutations, were in normal cells. So in fact, I really had to adjust my thinking here, because we could create and we could demonstrate that there was widespread mutagenesis in normal cells. And in fact, recently, because of the change in technologies, people have been starting to show that normal cells, before they turn into cancer cells, do have drivers, they have signatures, and in fact, they can even have little clonal outgrowths. So actually, this accumulation of mutations is happening in all our normal cells. So this was a real adjustment of dogma, because we've often been taught that these our cells, our DNA comes under attack from a lot of sources, and you can have a lot of different kinds of DNA damage, whether they're single strand breaks or bulky adducts or mismatches or insertions, lesions, and breaks. But we often have this enormous amount of repair pathways that help to fix a lot of this damage, and so that helps to keep the genome pristine. But that's clearly not the case, even in our normal cells. So I had the good fortune of working in the MRC cancer unit when I arrived in Cambridge. This is one of my colleagues, he's also a PI in Cambridge. And we were bouncing around this idea about why our cells tolerate such heavy mutagenesis. And so it turns out that not just in vitro, not just in the dish, that in vivo as well. So in humans, all our normal tissues have really quite a lot of somatic or acquired mutagenesis, including drivers, which are shown by these little black dots, thousands of passenger mutations, and may even have thousands and thousands of these little clonal outgrowths in all our tissues. It's actually very rare that a clone will transform into a dominant malignant clone and becomes a cancer, right? So because we have 30 trillion cells, and not everybody develops cancer. 1 in every 2 or 3 people develop a cancer. So actually, the occurrence of cancer is really quite rare. So how come we have so much mutagenesis? Are the DNA repair pathways not working properly, even in all our normal cells? And that seems unlikely, because we don't see any evidence of DNA repair abnormalities, because they produce very specific signatures. To the best of our knowledge, these DNA repair pathways are probably working normally. So perhaps it is our capacity for repair that has limits. It's not possible for our cells to just have a linear and unlimited relationship between the amount of damage and DNA repair, because DNA repair actually costs, right? It requires ATP, it requires nucleotides. And if there's too high a demand on a cell, then that risks cell death. So if we had DNA damage on the x-axis here, on this hypothetical diagram, and then wondered how the amount of DNA damage would affect the likelihood of, say, cell division. If the amount of DNA repair was unlimited, as seen in this blue line, then the higher the amount of DNA damage, the lower the rate of cell division, potentially to the point of cell death. So that's why you cannot have this unlimited relationship. By contrast, if there were limits, so you were allowed to do repair, but you were not allowed to do too much repair, as seen here in this turquoise line, I've gone for the Cambridge and Oxford colors, really not intentional, I think it just ended up like that. Then a lot of DNA repair could still occur, but there will be some damage that will be allowed and unrepaired, which will result in mutagenesis. So in effect, there's a trade-off. The cell prioritizes survival over genomic perfection. And that's why you still have quite a lot of mutations in your normal cells. This actually makes some sense, because the vast majority of the genome actually doesn't code for very much, right? Because coding sequences in the genome is only 1.5%, 2%. The rest of it's non-coding. So if you get bucket loads of UV damage, for example, sun damage to your skin, to the non-coding region, actually, who cares? It doesn't really matter. So we did perform some mathematical modeling to explore what would happen if you had cells that had unlimited DNA repair, here in blue, and cells that could still do repair, but on which there were some restraints. So this is cells in cyan. Let's see whether this works. Not going. Oh, it is. So in a situation where DNA damaging events are actually rarely deleterious, and most of the mutations are passenger mutations, then in periods of high damage, the blue cells that have unlimited repair, they can repair as much as possible. They see all the damage and they incur very high costs repairing all that damage to the point of becoming depleted and they die. By contrast, the cyan cells, they do have limits on repair, so they can do some repair, but not too much. And they'll just repair what's necessary only in an effort to survive the onslaught. And so that's why they're still around and they survive, okay? So given that 98% of the genome is intronic or intergenic, and the majority of mutations that arise as a result of DNA damage are unlikely to affect protein function, this hypothesis might explain why normal, healthy cells have such a high burden of mutagenesis, this permissiveness to mutagenesis, sacrificing genomic perfection over cellular survival. So this is just a hypothesis. This is early stages. We're trying to explore this idea, but clearly that concept that the genome is normally pristine, there's nothing wrong with it, and it's a high amount of mutagenesis is only seen in cancer is actually not quite correct. Oops, what's happened there? I was going to tell you a little bit about this, but I'm going to start running out of time, so I'm just going to move on and tell you a bit about how we use all the mutations in the genome to interpret cancer genomes in patients, bearing in mind what I've just shown you, which is that this idea that there's mutagenesis in cancer and in normal cells, and actually maybe some of the mutagenesis is absolutely fine. What we really need to do is identify the mutagenesis that's important for clinical reasons. And what's very important is to interpret all the information together and not just to pick or cherry pick specific drivers or specific signatures. Actually, you need to have all that information together in order to make sense out of what you're seeing. So I'm going to try to illustrate that with a patient case. So this is Joshua Barnfather. He's 30 years old. I have his permission to talk about him. And he is a patient who was diagnosed with a very rare genetic disorder called xeroderma pigmentosum type C. It's an inherited recessive condition, meaning he has to inherit an abnormal genetic defect from both of his parents to have the disease. And XP patients in the past, they tended to present like this, as you're seeing in the bottom right-hand corner. XP is a gene that's involved in a repair pathway that usually fixes damage from UV light. So if you get UV damage, when we get sunburn and we get pain, and that's part of the repair pathways coming in to try to fix that UV damage. And so patients with XP cannot fix UV damage. So what happens is they get a lot of sun damage. You can see a lot of freckling in some of these kids. Eventually they get sunburn, severe exaggerated sunburn, and some of them eventually die because of skin cancer from this marked UV damage. These days, patients are very well looked after by the various DNA repair clinics. They get meticulous photoprotection. They're all well educated, so they know about UV protection, etc. And they come to a regular clinic. So if they get little skin cancers, they quickly get removed. So these days, kids come up to the age of 29, 30. They live through into adulthood. So this is Josh at the age of 30. And in 2017, he presented with his violaceous lesion on the medial aspect of his left eyebrow. And it was diagnosed as an angiosarcoma, which is a rare cancer of the blood vessel lining. He had something called a wide local excision, so he had it removed and he had a bit of a skin graft here, but no other treatment. But in September 2017, he presented with metastatic disease in the submandibular lymph nodes bilaterally. He was given these treatments, and by September, by June, he clearly hadn't responded. In fact, he didn't respond at all. And he went on to additional trials to try to manage his disease. So he's got disease in his liver, in his pleura, and his lymph nodes. By July 2018, this man is dying. He has got disease in his cranium. It's extending into his brain. He's got it throughout his mediastinum. He's got bilateral pneumothoraces. He's bedbound and he's in hospital. And so things were really very bleak for Josh. This is his whole genome. As you can see, there's lots of red dots in it. He's got 800,000 mutations, and they're all C to Ts. You've seen this pattern earlier. Now, we hunted down his drivers. He had very boring ones called CDKN2A and P53. These are very boring drivers, and they don't give us any indication of whether this is an angiosarcoma or not. There are some drivers that we usually find in angiosarcomas. We didn't find them in his. But if we look at his mutations, his mutation patterns, you've seen this earlier. These tall red bars are what you see from UV damage. And to all intents and purposes, so that would be the UV signature, signature 7. To all intents and purposes, if you pass that through most cancer genomics people, they'll say this is a malignant melanoma based on this UV pattern. But remember, he is an XPC mutant, so he cannot fix UV damage. And remember that normal cells have a lot of damage anyway. You and I will have UV damage. He will have even more because he can't fix any. So actually, the UV damage in this guy is probably part of the normal spectrum for him. What's really killing him is this. So you can see this tall blue and pink bar come from another signature called signature 10, which is associated with mutations in a gene called polymerase epsilon. We couldn't find the mutation initially, but after some hard digging, we did find it. It was present in a very small fraction of cells, so it's called a subclone. And it was, interestingly, it's got a C to T pattern, which is this red bar. So we think that UV has hit the gene and unfortunately has created a driver mutation in a subclone. And it's this subclone in this tumor that is what's killing him, that's gone through all his organs and is what is costing him his life. Another reason why this was really valuable information is because Pol E signatures, Pol E mutations make a tumor sensitive to a specific drug called a checkpoint inhibitor. So potentially this guy could have checkpoint inhibition. He is at this point dying. So one of the ways that we have to try to prove that somebody might be sensitive to checkpoint inhibitors is to do additional protein immunohistochemistry, so testing of the presence of a protein. And when we did it in his primary tumor, that came back as negative. So that was a bit of a blow. But of course, remember, I said it was present in a small number of cells. So it might be present in just a portion that we simply haven't got a slice of. So at this stage, we said, do the staining in one of his metastases. And the reason why we were pushing so hard is because this is not standardized therapy in the UK. It's not been approved. So he would not be able to get this drug unless we can absolutely prove that he should be getting it. Thankfully, it did show positivity. So it's 60% positivity. At this stage, we have the scientific evidence to back him getting this treatment. But of course, this is not a recognized treatment in the UK, or rather not recognized, but not approved so far. And so the NHS wouldn't pay for it. This was a learning process for me. We had to go through what we called IFR applications. So you try to get funding applications through for a very specific reason. We put it in twice, and both times they were rejected within 24 hours. I have never seen so much grace in a human who was facing death. And he raised his own money. So he crowdsourced his funding for his treatment. This picture of him in the right-hand corner, you know, he's lost 20 kilograms. The reason his face is distorted is because his lymph nodes are so enlarged. And I remember some of the doctors saying it was too hard to look at him. But he was still smiling, as you can see in that picture. In the last moment, he got his drug. And I'm pleased to say that these are his images before treatment. You can see these horrible cancerous lesions in his liver, around his mediastinum. And this is him after 3 cycles of this drug called pembrolizumab. There's been significant reduction in the volume of disease and partial metabolic response. By the end of 6 cycles, everything had kind of disappeared away. So he's now walked home from hospital, and he is, as ever, is smiling. So this is an anecdotal story, but it, I think, demonstrates the value whole genome sequencing has for these sorts of scenarios. But that it requires a little bit more than just looking at the genome and reading it. It actually requires a bit of interpretation and thinking. We did have to consider the data, and there's a little bit of toing and froing and pushing and pulling in order to get the patient the right treatment. There's a picture of my kids running around on yet another holiday. Sorry, I've had so many holiday snaps, so thank you for putting up with all of them. I think progress is impossible without change. And, you know, you do have to be able to change your mind. Otherwise, you can't change anything in the world. And there's one thing I have noticed about myself is that, you know, I feel every time I feel like I've learned something, I figure out that I haven't quite figured it out. And so I've come to embrace that, and I actually rather enjoy the fact that I can chip away at an idea and evolve it, I guess. So I think embrace anything that comes your way would be one of the things I would say has been something that has been a guiding principle for me. So I'm going to end with my acknowledgement slide. This is my team. I'd also like to acknowledge the leadership of the International Cancer Genome Consortium, particularly the Breast Cancer Consortium, led by all those people who are named there. They had the foresight of doing whole genome sequencing at a time when whole genome sequencing was not the norm. I'd also like to highlight the collaborators named there for the stuff that I presented today. Of course, none of my work is done without collaboration. So there are many, many collaborators who are not named here, whose work has not been shown today. But there's many, many of those. And of course, this is my team. We hail from many, many different countries across the world. And we might have some job openings. So if anybody's interested to come and do projects with us, just give me a buzz. Thank you very much. I'm happy to take any questions if we have time. Thank you for listening.
Well, thank you very much, Dr. Nik-Zainal, for your really interesting talk and the beautiful story of how you managed to translate your work in DNA mutations to treatments for actual patients. So it's a really beautiful story. We'll now open the floor to Q&A. If you have any questions, please fire away.
They're stunned into silence. What day is it today? Today is Tuesday.
Tuesday, yeah.
Could I ask a question?
Yes, of course.
Which is, you showed us some fascinating work sampling one tissue, but then what should we do in the future? Presumably the early work came from sampling a tumor, but which bit of the tumor should you sample? And then you showed us, gosh, you've got to look at the metastases as well. And what about the healthy cells? How much do we now have to submit for analysis?
Yeah, what a great question. So in the early days, we were limited technologically by how well we could do the sequencing. And so we were taking massive chunks of tumor just to be able to get enough DNA to do all that whole genome sequencing. These days, this is work by others, not by my team. People in London, for example, people in the US, they have taken samples and then they have, cut them up into smaller pieces and separated them out and sequenced them separately. So we'll call that multi-regional sequencing of the same tumor. And from there, you can construct these phylogenetic trees to show what is shared between those different pieces and then what is unique to certain pieces. You can also do studies where you get multiple samples per patient. So if you have a primary cancer and then you have multiple metastases, you take samples from all of those and sequence them, and you can draw these complex phylogenetic trees. A lot of the early mutations that occurred, you remember right at the start, I showed how everything's a clonal outgrowth. Actually, a lot of the mutations in the clonal outgrowth, they will be seen in all the different samples. But in the individual samples, there will be some unique features as well. So I think it's still early days. I didn't have enough time to go into quite a lot of this sort of aspect of things, but it is very, very interesting. It's early days. I think we need to do better in terms of getting samples. So a lot of those early studies or the stuff I showed you was single samples, so single snapshots. We need to do more of the multiple snapshots or snapshots or snapshots over time. But that still just captures information about tumors. Your other point was about normal tissues. And the problem with normal tissues, the slight problem we have with normal tissues, is that it's a bit more mixed. It's polyclonal. So you have many, many different little clones growing up in our normal tissues. It's very difficult to see the mutations then. But increasingly the technology is improving such that we can start to see them. So I will say it's very early days. The stuff I'm showing you today, the stuff I showed you today, a lot of this work has really only started happening since 2012. So it'll be only 8 years into this field. Watch the next 10 years. I mean, I think we're going to learn a huge amount when we sequence normal cells. I didn't have time to go into it. There was one slide I showed where I said that some people were starting to sequence normal cells and they could see drivers and signatures. So some people are already doing that, and they can see a lot of this mutagenesis in normal cells, in the normal cell. So I think we will eventually have to adjust even some of our descriptions of mutations. So right at the start, I talked about drivers, right? I said some of these mutations are important for causing cancers. Actually, some people have already shown that when you sequence normal tissues, you've got a whole bunch of drivers too. So maybe they're not drivers at all. Or maybe some things we think are drivers are not necessarily drivers. They just happen to be there because they're common, or they confer a little bit of selective pressure for a while, and then after that, they don't confer any selective pressure. So, you know, your questions are very, very good questions. I think we still have a lot to learn, have to do a lot more sampling. Of course, it's going to cost a lot of money. So, yeah, I think it remains to be seen.
Thank you very much.
May I ask a question, please?
Yes, of course.
Well, thank you very much for the insightful talk there. I was wondering regarding, I think you mentioned a point about permissiveness of mutations in non-coding regions. And I was wondering in that regard, do you find more instances of, for example, translocations that involve that don't involve necessarily mutations within the protein coding region? And in that regard, do you have instances, for example, where you have something going on to the promoter of a very highly expressed gene, and do you see more of that than you do with protein coding mutations?
Fantastic question as well, golly. So, so substitutions and indels, you know, the small mutations, we do find that there's probably a few more of those in the non-coding intergenic regions than in the coding regions. And that's probably because the non-coding regions are more simple. They have less complex sequence. So instead of having a lot of AT-GC that's mixed up, they might have a lot of AT-AT-AT-AT-AT or GC-GC-GC. So there's low complexity. And when there's low complexity, your chances of having an error are higher. And if your chances of having an error are higher, then your likelihood of developing mutations is higher. So we find more subs and indels at higher frequency in these non-coding regions. But for translocations, for rearrangements where your sugar phosphate backbone breaks, actually we find those tend to be more frequent in coding sequences, or not in coding sequences necessarily, but nearer coding sequences. So more highly transcribed regions, busy regions. And I suspect it's, to get a rearrangement, it's a different kind of damage, right? You need to actually break that sugar phosphate backbone. And I think you need to have a lot more sort of traffic in the region, a lot more activity, whether it's transcription or, you know, just a lot more. So in regions like promoters and enhancers, you have a lot more activity in cells. So we do find that there is some variation in the distribution of the mutations depending on the signature. There are some signatures that are very extremely strong in some of those highly transcribed regions, and we think it's because of transcription in those sites. So yeah, it's a little bit variable from one signature to another. Generally, the rearrangements tend to be enriched in the sort of the more gene-rich regions than the subs and indels.
Thank you very much.
Pleasure.
Could I ask a question?
Sure.
So I guess this, I don't know, this may be a bit of an out there idea, but then like, if you were to sequence like, let's say normal tissue of patients before, and you know, use this kind of technique where you find out the rate of mutations of, you know, like, you know, to like HR or other kind of mechanisms, can you like estimate whether they'd be more susceptible to cancer this way? Or like Kind of more
I
as a
see.
diagnostic than.
Yeah. So perhaps use it in a kind of a predictive thing. So if you have more
Yeah.
of these mutations, could you be more likely to develop cancer? So in a
Yeah.
way, I think that environmental mutagens could help you there. So if you found that someone did have a lot of aristolockic acid signature, for example, in their cells, then you'll be there going, okay, hang on, why have you got that? Especially for the patients who say that, who claim they have no exposure, they clearly have some exposure they're not aware of. So I think, yes, I think in some instances it can tell you about exposure. And we assume that if you have a higher exposure, you have a higher likelihood of developing cancer, right? Because we know that smokers tend to develop lung cancers. It's not deterministic, so I don't think it means that if you have the signature, you will develop cancer. But I think if you had a lot of some of those signatures, then it's a bad sign and your chances of having cancer is higher. So I think you're right. I think there will be some opportunity to use it that way, but we'll have to do a lot more studies to try to figure it out, because as I said, I don't think not everybody who smokes will develop cancer, right? Only some of them do. So it's the same thing. And so if you use a signature in that way, and it's a good question, because I think some people will think about it in that way. I think we need to find out how to use that information responsibly to inform people without alarming them. But yeah, I think that potential is there. So mutagens would be an easy one. But yeah, HR and mismatch repair would also be good ones to use, because you know that if you have a HR pattern or mismatch repair pattern, that your repair pathway is not working, that's a bad sign. And so, even if it means you haven't got a cancer now, I think what it means is your risk of developing cancer is much higher than anybody else. So yeah, I think they could be useful. Absolutely. These are good questions I'm getting.
Hi, could I ask a question, please?
Sure.
Can I just say it's a fantastic talk? It's very interesting.
Oh, cool. Thank you.
I was just wondering, you talked about the benefits of doing whole genome sequencing rather than just targeting a specific gene. So you talked about how like the PIK3CA mutation is not present in all cancers. So I was wondering that would it be possible, rather than to sequence a whole genome, which is very expensive, to sequence a sufficient number of genes that the p-value of them all being there would be low enough that we could still confirm diagnosis?
So when you sequence, so the human genome, 3,000 million base pairs, the coding sequence, so just the exons, and there's about 20,000 genes. So the coding sequences make up only 1.5% of the human genome. So let's say we have, 5,000 mutations in our cancer genome. So we're going to use 5,000 mutations to characterize the genome. Now, when you're looking at just the exome, you're looking at 1.5% of 5,000. So, 50 to 75 mutations only. Suddenly your power gets really low. That's all the coding sequences. Now, if we just look at a few genes, that gets even smaller. People still do it though. So sometimes, and actually it's commonly used. So targeted sequencing is an assay which a lot of people use. It might be between 200 to 500 genes. It's called a targeted gene panel. And then people try to do signatures on them. The problem is that the power for accurate classification goes right down. But they do try to do it. So it's not impossible. People try. I don't know how accurate it is. I think it's well known that it's not very accurate. And of course, then what happens is that if your classification is inaccurate and you go into clinical trials or studies with inaccurate classification, you still end up with slightly nonsense results at the other end. So so I hear the concern is that whole genome sequencing is expensive and that we need to try to find ways of reducing that genomic footprint for an assay. And certainly people are trying. In fact, we are also trying to do that. Having said all that, a whole genome today can be done for less than £500. So actually, that's almost cheaper than doing a CT scan. So it's not that expensive anymore to do a whole genome. And by the time you guys come through university, the most limiting thing is not going to be the price of a whole genome. It's going to be the analysis and interpretation. It's going to be how you analyze the data, because you've got so much data trying to make sense out of it computationally is really quite expertise intensive. There's a lot of expertise required. I think that's going to be the main rate limiting factor for whole genome sequencing analysis and interpretation. But you know, that's why you have people like Google and DeepMind, you know, they come along and they make it really easy. So like the algorithm we have developed there on HR Detect, you know, it can be run like that. And that's what you want. You want, when you're a clinician's consultant, what you want is not to have to sit there and go through the whole genome. What you want is a report that comes out that says 99% probability of being HR deficient, you know, 85% probability of being mismatch repair deficient. You want the algorithms to have run over it and to report it back. And that will come. I think that will absolutely happen within our lifetimes.
Thank you so much.
Yeah.
Can I just ask your email, please? Would you mind?
Sure. It's snz@mrc-cu.cam.ac.uk.
Thank you.
Pleasure.
Thank you so much.
Pleasure.
Are there further questions from the audience? Actually, maybe I'll just ask a question first. Yeah, could I just ask, so currently most of the whole genome sequencing you've done are on cancers or which are known to have a certain mutation in DNA repair genes, or they are known to be caused by mutagens like smoking or UV light. But what about cancers which are driven by one or two driver mutations? For instance, like the classical CML, which is driven by BCR-ABL mainly. So how does the mutational signature look like for cancers which are driven by just one or two drivers?
So I've done a lot of cancer sequencing of breast cancers. They were not pre-selected. They were just breast cancers that had been sequenced that people had in the freezers, basically. There was no pre-selection. But the rest of the world have sequenced a lot of cancers as well. So, you know, there's not very enough time almost to kind of go through all of it, but a lot of people have done sequencing on a lot of cancers around the world. And a lot of that data is widely available. So you can go and look and explore things and try to find out what signatures are there. And in fact, I'm not sure that I'm able to do this, but I might show you somewhere that you can even look. So if I present now, entire screen, allow, can you see my screen?
Yep, I can.
And you can see something that says signal?
Yep.
Yeah, so this is our website that has a lot of information about mutational signatures. It's widely available, so it's open to the public. Anybody can use it. It's got all the information I was talking about today for you to explore. And this just tells you a bit about it. So it allows you to just browse through data through all cancers. You can even do your own analysis. You can actually upload data if you have data in it. So it's very simply organized. It's called Explore or Analyze. So click on Explore. You can look at cancers. You can go look at mutagenesis of environmental compounds, and you can look at mutagenesis caused by the knockouts that I talked about. If you click on cancer, you can look across all the different tissue types, and these are all the different signatures. So CML, I think, would be a myeloid leukemia. I don't know whether we have CML specifically. So it says here 38 primary tumor whole genome sequences analyzed. 6 substitution and no rearrangement signatures extracted. So these are all the different signatures that are in it. If you go to rearrangements, you don't have any because there aren't any rearrangement signatures. But, you know, this is signature 30. This is deamination of methylcytosine. These 2 signatures are supposed to be associated with, well, this one is supposed to be associated with bad outcomes. And this signature is due to platinum. So you can kind of browse information here. You can look for your favorite cancer, stomach cancer. You can see mismatch repair, deamination, Applebeck, signature 18, Applebeck's again. So it's possible to browse all of this data. Now, I didn't sequence all of these cancers. Everyone else in the world did, but everybody shares data so that we can all analyze and share processed knowledge and information together. Does that make sense?
I think it's a fantastic database.
Cool.
With
Please play around with
lots
it. And we're always happy to have feedback. It's quite a new database, so there's still a couple of bugs in it. But yeah, if any, you know, and you can even download the data. So if you're exploring something, you've got your favorite tumor type, and then you want to download the data to play with it yourself, you can. So you'll be able to do that. And then there's a help desk with it. It's run by two guys in my team, Yan and Scott. And they are fantastic. They always respond very, very quickly and very, very effectively, and they always listen to try to improve the website as well. So, yeah, so give it a run, and if you can think of any suggestions, please let us know so that we can improve it.
So, I mean, given this huge amount of database, what do you, and all this research regarding DNA mutations, what do you think is the main hurdle to implementing this sort of whole genome sequencing methods of looking at the whole mutational signature rather than individual genes? What do you think is the main hurdle to implementation in the clinic?
So I think a lot of people are still kind of focused on doing targeted sequencing. So they haven't done whole genome sequencing in a clinical context. They've done it only in an academic context. A lot of the stuff that you're going to see in that Signal website is all academic. So there's very few in the way of proper clinical studies with whole genome sequencing. So the main barrier to it is mindset. I think a lot of people still think it's very expensive, and a lot of people are very fearful about how to do analysis and interpretation. So I think the first one is a myth now, because it's quite cheap to do whole genome sequencing. But the second one is still true, that the analysis and interpretation expertise required is massive. So at the moment, we're trying to make that a little bit easier in my team. But of course, it takes resource, right? So, you know, it's a bit like when you go exploring with a machete to find a new place. You find a new place is all very exciting. To build the highways and the roads to be able to get to these new places, you need other people to come along and make it fabulous. And at the end of the day, we are explorer scientists, you know, and we need to get in the experts to come and do the bioinformatics to build those super highways. And that costs. One, nobody really prizes that, which is sad. But it is important, because otherwise these things don't get translated. So we are starting to look into how to make that a little bit more affordable for a research team and to get funding, basically. And then this is how sometimes people end up spinning out companies, because you can get investors to come in, you know, pay for that sort of thing to be done, rather than having a research group doing it. So it's actually a really good question. It's a really exciting time, because we're absolutely at that edge now, trying to figure out how do we make this go into clinic quickly.
So it's really about building this bioinformatics team to set up a system and a pipeline to make it easier for clinicians to use this in hospitals. Yeah,
I think so. I think in the end, that is what it comes down to. In this day and age, we share knowledge and information so quickly, right? So everybody knows all the difficulties associated with whole genome sequencing, because it's all being shared. But if you go back 5-10 years, if we look at the development of MRI scans and CT scans, I mean, none of us know anything about how they were built and developed, but there are MRIs and CT scans in every hospital, and everybody gets their images done. You know, nobody questions the hardware and software, but what we learn how to do as medics is how to read the CT scan images, and that's the same principle. We have all become so fixated on the bioinformatics of how things are done that I think that is scaring us. But actually, as clinicians, all we need to learn how to do is how to read the genome reports at the end. And you did that already today. When you looked at those whole genomes, you could instantly see how one genome was different to another. That is the clinical teaching that needs to happen in order for us to train the next generation to be able to read cancer genomes.
Thank you so much. Are there any other further questions from the audience?
Good.
Yeah, right. I think we can wrap this up then. Thank you so much for your really insightful talk, and thank you everyone for joining us today. If you're really interested in this sort of biomedical talks, we actually have another talk next week, and it's by Dr. Sarah Teichmann on single-cell RNA sequencing. So more sequencing stuff.
Fantastic. Thanks so much for the invitation. It's been a real pleasure.
Thank you so much for joining us.